Hello, everyone. My name is Sarah Dilorio. I'm an MD PhD student in the Longacre Lab, and today I'll be presenting a single-cell RNA sequencing meta-analysis of mouse tendon cells. As you probably know, tendons are very commonly injured tissues. They account for over 30% of all musculoskeletal consultations. Tendons have a very low vascularity and cellularity, which means that after injury, they have a very low regenerative capacity and don't heal very well. One side effect of this is that they can often heal with fibrosis or adhesions, which is different types of scar tissue that can impair range of motion and reduce the function of the tendons. The specific cell subtypes and signals contributing to this fibrotic response are not well understood, and so this is something that we're seeking to identify through our project. For the methods of this particular study, we looked at all publicly available RNA sequencing data sets through NIH Geo and other similar platforms. We had a particular selection and criteria to look at just mouse studies so that we could compare it to our own mouse work. We also looked for adult tissue rather than any embryonic or young tissue. And we also looked at anything that was droplet-based sequencing technologies only. From here, we found 9 mouse data sets that met these criteria, which we then integrated and performed cell identification through clustering using Seurat version 5. We also compared the uninjured and injured conditions, so basically where the tissue came from in the mice, and then also we subclustered fibroblasts and tenocytes to look at subpopulations. Moving into our results in our first top figure here, we found 15 transcriptionally defined clusters when we looked at all cells from all studies, the most interesting of which being the green and pink cluster here, to two identified clusters, which were both the fibroblasts and tenocytes. And so we took those clusters and created a separate data set, which we then subclustered for additional analysis, where we identified 3 fibroblastic clusters and 1 tenocyte cluster. We were excited to see that tenocytes and fibroblasts clustered out separately, indicating that even though they're very similar cell types and that they maintain extracellular matrix and produce collagen, there's enough difference in gene expression to distinguish them. We then looked at comparisons between injured and uninjured conditions, first in the overall cluster and then again in our subclustering. In the overall clusters, we saw some differences in these fibroblast and tenocyte clusters, as well as a pretty big difference in our myeloid cluster, which was primarily immune cell based. We also saw that in the subclustering analysis, the fibroblasts, especially this red and blue population, clustered almost entirely to the injured condition, whereas the tenocytes were almost entirely in the uninjured condition. And this is consistent with existing literature that shows that fibroblasts are typically more responsive for the scar formation, while tenocytes are more responsible, for the regenerative kind of properties of responding to the injury. In conclusion, we saw that tenocytes and fibroblasts are computationally distinguishable, and we saw that the fibroblast populations were primarily present in the injury condition, while tenocytes were in the uninjured. These data will inform our future studies into the cellular and molecular mechanisms of tendon adhesions and help us understand the contributions of both these fibroblasts and tenocytes. Looking more specifically into our planned future directions, we hope to perform our own single-cell RNA sequencing of tendon to look for our own similarities to the findings that we saw with the publicly available data. We also hope to compare this publicly available data between different time points and types of tendon, since that data was available to us and it's a further analysis that we have planned. And then we also want to perform in vivo experiments to validate the markers that we find both in this publicly available data and then also in our own personal sequencing. I'm excited to discuss this further at Holman, and I welcome any questions you may have. Thank you so much for your time and attention.